omega-3. I just was sitting here thinking, I've been thinking about omega-3 for almost 20 years, which I'm aging, so. But when I started out talking about omega-3 and learning about omega-3, you had to spend a lot of time explaining what omega-3 was to people. And nowadays, everyone's heard about omega-3, everyone has some knowledge, but I still find that people don't necessarily really understand omega-3. So today's talk, I'm going to talk a little bit about the how, what, why, and where of omega-3 fatty acids. I am a little bit different than most of the speakers today. I think I'm the only one wearing a tie today. And as a male, I like to keep things simple and think about one thing at a time, so I'm going to focus really on just omega-3. So when we start off with omega-3, I'm going to go back and just talk a little bit about types of fats and fatty acids and my perspective on these. So when we look at fatty acids, these are some kinds that we see up here, but one of the big characteristics is they're hydrocarbons. So what am I talking about? I'm talking about the atoms and molecules, but really we're talking about a lot of carbon molecules, a lot of hydrogens, very little oxygen. And in essence, they're very similar to petrochemicals, so gasoline, etc. There's a lot of common knowledge, and fatty acids are a form of, they do a little bit of tweaking to them and they can make biodiesel of them. So one of the things about them is they have a lot of energy in a small amount of space, and you don't need to store water with them. So typically we think about fat in terms of energy and being fat. But when it comes to omega-3, it's a little bit different story. So one of the things when you look at these fatty acids, we have saturated fatty acids. And the thing about saturated fatty acids, the word saturate means that there's no carbon-carbon double bonds. And then we can look at the monounsaturated, so mono meaning one, we see one carbon-carbon double bond that puts that kink in it. And then we have things called polyunsaturated fatty acids, which have more than one carbon-carbon double bond and make them even kinkier. Okay? So, and then at the bottom we have the 18,3 and 3. So the polyunsaturates, we can divide them into two classes, the omega-3 and the omega-6. And how do they really differ? If you first look, you might not notice, but the big difference is this one carbon-carbon double bond. So it's in the 3 position from the 1 tail. I'll talk a little bit more. Now, what do these mean? What do these carbon double bonds mean? Well, if I gave you a big chunk of saturated fat, say something like butter or lard, it's a solid at room temperature. But if we had, say, a polyunsaturated fat, we're talking about things like corn oil, safflower oil, and that's a liquid. So in our body, they have big differences in how we handle these fats. A little history on omega-3 fatty acids. A lot of times this is a 1964, they claim it was first used by Ralph Hallman, but I actually found a paper where he used it in 1963. So omega-3 fatty acids is a product of the 60s, like the baby boom generation, everything great came out of the 60s. So Ralph was a lipid biochemist / nutritionist looking at essential fatty acids. So he was looking at the metabolism of linoleic acid, which sometimes we call 18,2 omega-6, and linolenic acid 18,3 and 3, or omega-3. Now, when Ralph was looking at these fatty acids, we were either calling them linoleic or linolenic, and this omega-3, omega-6 nomenclature the names didn't exist. If we looked at them in terms of chemical structures, we'd have things like 18,2, delta 9, delta 12. Okay, and then the N6 one, or the omega-3, was 18,3, delta 9, delta 12, delta 15. So this nomenclature is just indicating what position the double bonds are from this end of the molecule. Now, with mammalian physiology, all the business end of the fatty acid is at this end. What am I talking about? Well, if I want to use this for energy, or if I want to make fat in my body, I'm making it at that end. So naming everything from this end sets up a situation where these numbers will constantly change and you can't see a family. And that's where Ralph came in. He kind of noticed this behavior. Okay, because when you get into the really long fatty acids, it gets really crazy. So we have a 20 carbon fatty acid with 5 double bonds, and it's like delta 5, delta 8, delta 11, 14, 17, etc., etc. Over here we have, again, it gets even worse. We have docosahexaenoic acid, so 22 carbons, 6 double bonds, all kinds of different positions. and nothing in common between these two, even though they're both omega-3. So what Ralph Homan did is he decided to name them from the dead end, the metabolically inert end, or the methyl end, or the omega end. The alpha end is the active end, and then the omega end is the delta end. So he would say, if I name them from that end, then an omega-3 is always an omega-3. And then we have these family of fatty acids. So just side note, docosahexaenoic acid is my favorite fatty acid. It's the long chain omega-3, and I'll talk a bit more. And not just because it's the kinkiest fatty acid. Okay, so after Ralph came up with these names, then we have things like EPA, eicosapentaenoic acid, which is 25N3, 18,3N3, the DHA, 22,6N3, and it was easy to tell that these were fatty acids in the same family, and we could convert them back and forth in our body. And really, Ralph Homan did set up things like this, understanding how we metabolize fatty acids. Okay, so this is a complicated picture. I expect undergrads to memorize it, and there'll be a quiz. I'm not going to make you do that today. So what I really want you to focus on is these three. Okay, so this is the 18,3N3, the 25N3, and the 22,6N3. So why am I focusing on these three? Well, these are the three main forms that we eat in the diet. The 18,3N3 is the plant source. So sometimes we find this in flaxseed oil, etc. The 25N3 and the 22,6N3, those are the long chain omega-3 fatty acids. Those are my favorite, again, because they're so kinky, but I'm going to talk a bit more. But what I want you to notice is we can convert that plant source into those longer chain ones that we find. But notice how complicated it is. Lots of different steps. And at some of these steps, we've got competition. We've got two different molecules fighting for that metabolism. So while we can convert plant-based omega-3 into those longer chain ones, we don't do a very good job of it. Okay? Because at the top, a lot of those 18 carbon fatty acids actually get used for energy, and a few of them will make it into those longer chain fatty acids. So typically the best source is often preformed EPA and DHA, which is typically found in things like fish. So omega-3 deficiency. Whenever we talk about nutrition, we hear about deficiency, and you'll hear, oh, North Americans are omega-3 deficient. Well, by the strictest sense of the word, there's hardly anybody who's omega-3 deficient. Again, Ralph Homan was actually the first person to observe someone who was omega-3 deficient. It was a 7-year-old girl who had a gunshot wound to the stomach. So all her feeding, she had intravenous feeding, and the fat they were using to feed her was safflower oil, which is loaded with omega-6 but no omega-3 fatty acids. Over a period of time, she started to develop neurological and visual impairments, which were reversed when they switched her over to a soybean oil, which had a little bit of omega-3. So a little bit of omega-3 will prevent these kind of neurological and visual impairments, which is kind of true of the classic term deficiency. But we've kind of changed requirements from preventing deficiencies to trying to optimize our health. And that's where omega-3s, maybe we don't have to worry about preventing deficiency, but if we want to optimize our health, especially with aging, maybe we need to talk about eating more. We confirmed some of this work in rats. So this relationship, Allison showed us some data about what people think about omega-3, and I was surprised that brain mental health was not listed, because it is one big area of research. Typically it's in developing infants, so this is actually some work that I did during my postdoc. Those are baby rats. We hand fed them and hand reared them, and you have to encourage them to pee and poo, so you had to hand encourage that. This is after 10 years of university education. I was peeing and pooping rats 4 times a day for 30 days in a row, which was lots of fun. But But with this model, we were able to control the amount of fat they ate because the maternal breast milk will have lots of omega-3 in it. So we could give them custom milk to look at exactly what the fatty acids did to their mental development. So I'll start the one video right away and then explain the study a bit more. There it is. So this is a water maze test. There's a hidden platform. And the rat's actually been trained to know where that platform is. He's been exposed to this before. He's been shown where the platform is, and there's visual cues in the room where he can look at and try to determine. So this rat was fed omega-6 without any omega-3. And we can see he's looking for the platform. He's been looking for a while. Oh, there. No, not going to find the platform. So we can see that this rat's really spending a lot of time, and it doesn't seems like it's just a random, not really finding the platform at all. Eventually we're going to see that it almost looks like it bumps into the platform by accident. And there it goes. And then on the other video, I'll start the other video. This is the rat fed DHA. So the rat will go in. Here goes the rat. Swims straight to the platform. Now the one big aspect about omega-3 that everyone seems to know about is heart disease. We got a picture of a piece of salmon in the shape of a heart. Allison's survey shows that everyone considers omega-3 with heart disease. So what's the history with that? Well, it comes out of the 1970s when fat in general was considered bad for heart disease, caused heart disease, fat was bad. But what happened was there were some Danish physicians. So a lot of Danish physicians, when they completed, will actually do some time and go up to Greenland. Greenland is still officially a part of Denmark, but they would go and do a rotation up in Greenland. So two Danish physicians, Bang and Dyerberg, went up to Greenland in the 70s, and they knew that fat was associated with heart disease, but they also knew that Greenland Inuit ate a lot of fat and very little fruits and vegetables, but for some reason heart disease wasn't affecting them. I was fortunate to meet Dr. Dyerberg. Dr. Bang had passed away before, but this was me at a conference in Japan as a PhD student, so it's kind of like meeting Elvis or Wayne Gretzky. For me it was anyways. I know most people won't care that much, but for me it was very exciting to meet Dr. Dyerberg. So what we see is the Greenlanders, despite eating lots of fat and very low fruits and vegetables, very few of them die from heart disease compared to Canadians and Americans. So eating lots of fat, no fruits and vegetables, not dying from heart disease. What's going on? So they started to look at the diet, and while they're eating lots of fat, it's a very different type of fat. We can see they eat lots of fish, which we typically associate with omega-3, but in addition to the fish, they eat a lot of marine mammal, so seal, whale. They actually eat more marine mammal, okay, but the marine mammals are actually eating fish, so they're getting lots of omega-3 from marine sources. So what kind of fat do humans eat anyways? Okay, I like to call it the big four. Any kind of civilized society typically eats about 90% of their fat comes down to four main fatty acids. 16O, palmitate, a saturate, 18O, another saturate, 18-1N9, and then 18-2N6. It typically makes about 90% of any kind of modernized society's fat intake. We can see the Greenland Inuits very different, only about 50%, so they're eating a very different thing. But then the other big difference here is the EPA and DHA, the long-chain omega-3. We can see the Greenland Inuit are eating about 13% of their fat, whereas Canadians and Americans are eating about 0.3%. If we look at a modern society that eats omega-3, we can look at the Japanese, who are eating about 2 grams per day, and typically they have less heart disease as well. How many people have heard of 369 omega supplements? Lots of people have heard of them. And people ask me what omega-369, should I take omega-369 or should I take omega-3? Well, 40% of the fat you eat is 9. Almost 20% of the fat you eat is 6. So do you really want to be spending more money on 6 and 9 in a pill? To me, it's a marketing ploy. So how does omega-3 work in heart disease? There's actually lots of different mechanisms. And this has changed over time. When I was a grad student, it was all about lowering the level of triacylglycerols or triglycerides in your blood. But there was a shift towards preventing sudden cardiac death. So we can see with the sudden cardiac death prevention, a little bit of omega-3 makes a dramatic difference, but then eventually it plateaus. Whereas the triglyceride lowering seems to be a linear, the more you eat, kind of the better type scenario. But then you get other things like heart rate lowering, potential blood pressure lowering, but then you get a risk of increased thrombosis with super high levels of omega-3. So a lot of the guidelines that they're making now based on how much omega-3 should I eat are based on this sudden cardiac death ratio. So those are the things that it prevents, but I want to talk a little bit about how it actually works in the body. So this is a diagram of basic fatty acid metabolism in general. Okay, and I've got energy and fat storage, typically the things that people think about fat. We use them for energy, we use them for fat storage. The big circle here, this rectangle, represents a cell sorting them. But where I get excited is about omega-3 gets excited, is the actual makeup of your cells themselves. So your cells themselves are little, basically, bags of fat. Like, not bags of fat, but the bag itself is a fat. Right? The compartment containing the cell is a lipid bilayer, and there's fatty acids in there, and that's where omega-3s do their magic. Why do we have different colors? These little circles represent different types of fatty acids. Too often they get lumped together as fat in general, but I think we really need to think of them as separate little molecules. I like to think of them as a bowl of Smarties. Not everyone likes the same color. Some people love the red, and we have biological processes that prefer the red Smarties or the red fatty acids, and we'll use them exclusively. And we see some of that with our cell membranes. So this slide, I'm just talking different, so the fat we eat can go into different places, and the type of fat will go into different places. So adipose fat, or the fat that I gained a lot since my PhD, sitting here right now, tends to have this 16 carbon, 18 carbon fatty acids. And these are the ones used for energy. You know, adipose is storing energy, so it makes kind of sense that this is the type of fat that you would store there. It's a solid at room temperature, white, right? But those cell membranes, those cell lipids, we see that, yeah, we have an 18O, but then we have a position where we have these long-chain kinky fatty acids. And that's where the omega-3s like to be, in that cell membrane itself. So once the omega-3 gets in the cell membrane, so we're looking at the fatty acids are being these parts of the cell membrane. So they get right into your cells. And once they're in your cells, they can do all kinds of things. They can be used to communicate with different cells, okay, as signals. They can affect the proteins in the membrane and the things that get through the membrane, and they actually can make the membrane more fluid, right? Think of it, if you had lots of saturates in your membrane, they'd be solid like butter. If you had omega-3, the kinky ones are going to be more flexible. And then they'll also direct all kinds of things going on inside the cell. A lot of these, again, kind of like communication signals within the cell. And this can affect things that happen immediately, but it can also express which genes are expressed at different times, so we can change gene expression. So there's all kinds of things that omega-3s do within the cell. So I'm going to finish the presentation talking a little bit about some research that we actually did here a few years ago. So one of the things that we talk about is how much omega-3 do people need to eat, but at the same time we have to figure, well, how much do Canadians actually eat? We don't have a lot, we have a little bit of data, most of the data on omega-3 and what people eat is usually done at university students, because university students are close to where we do our research. You know, you wave a couple of dollars at them and they'll jump for that dollar, so they'll do the research for you. So at the time we had very little data on what older communities, retirement communities, how much omega-3 they eat. So we went in to Winston and looked at how much they ate. So what you're looking at here is actually data where we actually collected the food, took it back to my lab. So we did a food duplicate. Every time, if a person ate an apple, we took an apple and put it in a hopper. If they ate a tuna fish sandwich, we took a tuna fish sandwich and put it in the hopper. And we brought it back to the lab and then analyzed it to see how much omega-3 everyone was eating. So every bar you see here is actually an individual participant. So we see a neat pattern here that the average was about 121 milligrams per day, which isn't too bad, it's not great. But what we really see is the average is not really a good estimate here, because there's a lot of people eating very little amounts, and we've got 3 people eating a lot. So the average is actually inflated by those 3. So we have to look at what we call the median as a better representation, and we drop down to 53 milligrams per day. Now, I'm always a little bit cynical about dietary assessment data. If you ask people what they eat, people tend to be like 3-year-olds when you ask them what they eat. Are they eating healthy? Oh yes, I'm eating healthy. But then you find the peas stuck under the mashed potatoes or something, right? So you always have to be careful with dietary assessment. But the other problem, that's why we did the duplicate food collection. But with the duplicate food collection, we're only collecting food for 3 days. And we do know that omega-3 is very variable. We're not eating fish, we're not a society that eats fish every day. We're not Japan. So people will eat fish on Friday, the Catholics will come out and eat their fish on Friday. Maybe once a month someone will eat salmon. So it's very tricky to assess omega-3. So one thing we do in my lab is, can we assess nutrient status by looking at people's blood? So when we look at people's blood, you have to kind of think, well, if we're looking at blood, what are we actually looking at? Okay, so people here probably getting blood samples before. They'll take the blood. Typically, they'll look at that plasma, the fluid component, but there's also a red blood cell component. So when we talk about fat and plasma, this is the kind of structure. We're talking about lipoproteins that have like a single layer membrane and some triglycerides inside. But if we look at the red blood cells, they have cell membranes like our cells. Okay, so the red blood cells might tell us more about the actual cells in our body. This might tell us more about what our liver's doing to the fat. So what we actually like to do is look at whole blood. Look at it all and see what it looks like as our kind of first screening. So this is the data from the blood from people at Winston. And we see we're expressing this as the percentage of omega-3 long chain in relative to the omega-6. So we can see the mean is actually 28.8, the median is 28.3. So we can see some of that variation is gone, that there is more of a median, and biologically we maintain it. We see individuals again for each bar, so we have a bit of a range, but it's not nearly as bad as it was before. Now, what does 28 mean to you? A lot of people, there's a lot of talk that, we should be looking at 50% omega-3, which I think is a little bit high. I think 40% is a really good number to be at. I think if you're a Canadian, being over 30% is fantastic, given our cultural habits. But this number, this average of 28 is really good actually, compared to university students typically fall about 20% average. So we'll see university students with even 15% omega-3. Now, is this high enough to be associated with cardiovascular disease reduction? I think it's on the way, but I think we could increase the levels of omega-3 for more benefits. Okay. I need to acknowledge the actual people who did the work, grad students and technicians in my lab. I'm the song and dance man, just giving up the presentation. And then all the sources of money, including the RIA and AHA. And I have to thank people like Hillary, who do all the grunt work, and then I just come in and give a talk.